Okay, I think it's time to get started here. So thank you for joining us, where we plan to highlight our metabolomics capabilities and explain how they can be accessed in order to advance your own research. And it's our hope that the presentation will provide an opportunity to get answers to your questions, whether they're specific to metabolomics or more general about the process of applying to become a user. Questions will be collected throughout the presentation, and we'll be collecting them through the chat function. So feel free to just type those in as we go along. And we'll take some time at the end of today's presentations to address these questions. And then we'll also capture these questions and make the answers available online after the fact for those folks who are not able to attend live. First, though, a little bit about us. The Joint Genome Institute is a U.S. Department of Energy user facility located at Lawrence Berkeley National Lab. And we're a world leader in genomics of plants, fungi, algae, complex environmental microbiomes. And we help researchers characterize how these organisms in these communities carry out complex biogeochemical processes, as well as how these organisms can be harnessed for sustainable biofuels and bioproducts. So again, we're focused on projects that are related to biogeochemistry, biofuels, bioproducts. So things that focus on primarily biomedicine or agricultural food crops usually aren't appropriate for the type of proposals we work on. So we have a number of different resources, capabilities, expertise, and in the areas of DNA and RNA sequencing, DNA synthesis, data analysis, and of course, metabolomics. These capabilities are provided to researchers for free through a competitive peer review process called the Genome Community Science Program, or the CSP. And this is important. In contrast to other funding agencies where accepted proposals are awarded funding, when it comes to JGI, successful proposals are awarded free access to these resources and capabilities. And we help you carry out the proposed work. So we have a number of different calls throughout the year through the CSP, and you can see the latest dates here. The next one is the functional genomics call in January 31st, and after that, our new investigator call. And metabolomics is one of the capabilities that can be requested in these calls. So as I mentioned before, today we hope to provide a better understanding of our metabolomics capabilities and how you might incorporate these or incorporate metabolomics into a future proposal in order to advance your own research. And we're going to start that by having a brief overview of the technology by Trent Northern, a senior scientist at LBNL and head of the JGI's Metabolomics Platform. And then after that, we're going to hear from Jennifer Mortimer about how she's been utilizing metabolomics to support her research at the Joint Bioenergy Institute. And then finally, we'll spend some time answering some of your questions at the end. So again, I encourage you to submit these questions in the chat bar as we go, and we'll do our best to address them. So with that, I'm going to turn it over to Trent Northern for an overview of the metabolomics program.
All right, thank you for that introduction, Rex, and thank you all for joining today. It's a pleasure to have you here and to tell you a little bit more about the capabilities that we have at JGI for performing metabolomics experiments. All right, and as Rex said, I'm the Metabolomics Program Science Lead. And just briefly, the mission of the Metabolomics program is to effectively integrate metabolomics with DNA sequencing and synthesis to provide users with new functional insights into DOE biological mission areas. And we have three different focus areas: functional genomics, environmental interactions, and secondary metabolites. But as I tell you at the end, we work on a wide range of particular applications. We have two core products. One is, and this really is based on the chromatographies that we use to separate the metabolites. One is for polar metabolite analysis using liquid chromatography, tandem mass spectrometry. And then the second is analysis of polar metabolites, again, using LC-MS. And as Rex mentioned, you can request metabolomics as part of any of the JGI calls. So any of these ones that I have listed on the right, and Rex briefly went through the deadlines for applying for those. So just in thinking of JGI metabolomics, we're really this next generation genomic sciences center, and you can use sequencing and it gives you a hypothesis about the function of a gene, but metabolomics is a way of getting at the functional reality. And so we take metabolites from a variety of different biological matrices. We separate them using some sort of liquid extraction typically, and then we analyze them using tandem mass spectrometry. So that means that we profile the ions as they're coming off of a HPLC column, and then we fragment those ions to get additional structural information. And then we use advanced cheminformatics and bioinformatics to integrate that with other types of information that you have, which is typically something like transcriptomics or proteomics or genomic information, so that you can really understand the function of your system, whether that be a microbial community, an environmental sample, or an isolated organism. And critically, JGI has a lot of other capabilities that you can put together as part of a CSP with metabolomics. And I just wanted to mention, for example, the JGI synthetic biology program, which would allow you to, say, have some genes synthesized so that you could test the function of a particular protein or make mutants to really get at causal mechanisms for these type of biological questions. So we have two different types of data analysis. The first is targeted analysis. And when we say this, it basically just means that we're using analytical standards or the measurements of analytical standards to annotate the ions that are in your sample. And so this is what we would consider gold standard identifications. So here we have this tandem or this two-dimensional plot that's showing on the x-axis the retention time as the ions are coming, or the metabolites are coming off of the column. And then in this y direction, they're being separated by their mass to charge ratio. And you can see they look kind of like HPLC traces, right? But this is what you get when you do mass spectrometry. You see that there's so many different metabolites coming off at the same time. And so you really do need this mass to charge separation to resolve them. And then for each of these, we fragmented. And so the fragmentation basically breaks the molecules into little bits. And then we compare that with our standards library. And so through comparing the types of fragments that we see, the retention times, the exact masses, the isotope distributions, we can get really high confidence identifications of those metabolites. And then we can also look at relative quantification of those ions or these metabolites across your various treatments. Now, we have over 4,000 standards that we use for annotating metabolites, but that's a drop in the bucket compared to all biochemical space. And here's just an example. This is another one of these two-dimensional plots where we have retention time as it's coming off the column on the x-axis, and then the mass to charge ratio on the y-axis. And we see all of these little blips, which we call features. And this is just LB media. And you can see that there's thousands of unique features here. So how do we approach this? Well, this is where we use a different type of data analysis, which is called untargeted metabolomics. And Ben Bowen, who's on as a panelist here, has developed extensive capabilities for doing both targeted and untargeted analysis. So for these unknown metabolites, we don't have standards for those. And oftentimes there aren't fragmentation spectra available for them. So Ben and Changul Pan developed software that uses chemical structures to predict fragmentation. And he's assembled over 200,000 known naturally occurring metabolites. And for most of those, he's been able to predict their fragmentation spectra. And so we can use that to annotate signals that we get from the metabolomics data that we don't have a standard for. He's also grouped all of these metabolites into a chemical network that is arranged based on their chemical structures. And so we can use this information to basically see what neighborhood a molecule falls in based on its predicted fragmentation spectra, and then annotate that by its nearest neighbors. And so this is what Ben is calling Pactolus, this overall capability. One of the challenges here is that maybe just like a gene blast score, you get multiple annotations for a given metabolite with different confidence in those. And this can be quite laborious to sort through them. So since we almost always have genomic information, Ben and Owner have developed software called Magi, Metabolites and Genes Integrated, that uses a carefully curated chemical reaction network to link together enzymes that are annotated from genomes or metagenomes or any type of sequence information. And then these Pactolus identifications, where we've basically taken guesses at what's the most likely identification of those compounds. And so we can put those together using Magi. So you've got to calculate a consensus score. So in one case, you might have a high homology score for a particular enzyme that participates in a reaction, but we don't see any evidence for this in the metabolomics data. So that would get a low score, whereas in this case, we get a high homology score for a protein that participates in this reaction, and we also see strong evidence for that metabolite. So this is a capability that we have for linking together any kind of omics data with our metabolomics. Another capability that we've developed and really worked with some of our users, in this case, Candace Swift and Michelle O'Malley at UCSB, is to use GNPS for relating metabolites together that we maybe don't have standards for. And this has now become a routine part of our untargeted metabolite analysis pipeline. And we release data through this software or this platform called GNPS. And what it does is it clusters together metabolites based on their fragmentation patterns. And so you might have a standard for this metabolite, but it clusters, just imagine that it's a peptide. And all of these other metabolites cluster with it because they're closely related peptides. So this is a way that you can kind of get an idea of what other metabolites in your compound, in your samples are based on the annotations of metabolites that cluster with them. I'm excited to tell everybody that we just got a brand new mass spectrometer, the latest technology. So we've been using Orbitrap mass spectrometers since the beginning at the JGI Metabolomics program. But now we have the first one that was really designed for metabolomics. And it can do things like perform MS to the N experiments. So we can not only fragment a molecule, but we can keep fragmenting the fragments of that for maximal structural information from those. And we can go out to 10 levels of fragmentation. Also has super high resolution for looking at stable isotopes and getting chemical formulas. And it allows us to do recursive experiments where we can do multiple injections and ignore all of the ions that we've detected in the previous injection, so we can get more comprehensive coverage. So we're really excited about this new capability. So together, our program, along with all the other programs at JGI, has extensive experience in studying all sorts of different applications. So for our program, we have done extensive work on cell and tissue metabolite analysis, DOC analysis, soil metabolite analysis, stable isotope analysis, lipid analysis, analysis of secondary metabolites and pathways, synthetic biology, using metabolomics for gene annotations, genome annotations, as I mentioned, integration with transcriptomics and genomics and proteomics. And then one application that is a real focus for us is what we call exometabolomics. And this is where we're looking at the metabolites that are in the environment and how different organisms transform them so that we can hypothesize metabolic interactions between, for example, plants and their microbial hosts. And we have a large number of publications on these various applications. And I think one thing that makes the JGI user program a little bit unique is that we're really deeply integrated with the user science. So right from the very beginning of having a proposal awarded, we have an initiation call where we talk through your science, and potentially we can suggest ways to improve your experimental design. We almost, if it's a new application or we have any kind of concerns, we will perform a pilot experiment to save everybody the time of going through and performing a huge experiment only to find out that there was some problem with it. And so these pilots are really aimed at making sure there's not some, we've got enough sample, we can detect the metabolites you're interested in, so it de-risks the whole experiment. And then we're involved in interpreting the results, and most of the time involved in helping prepare publications. And so Catherine is the Metabolomics Platform lead, and she has tremendous experiments with metabolomics too. And then Ben Bowen is really involved in the back end with helping advise on data analysis approaches, ways to do data integration, statistical methods, and again, also helping figure out how best to present the data in a publication. So with that very brief introduction, I want to introduce Dr. Jenny Mortimer. So she is a staff scientist here at Lawrence Berkeley National Lab. She's the Deputy Vice President of the Feedstocks Division at the Joint Bioenergy Institute in Emeryville. She got her bachelor's degree in biology at the University of Bristol, and then a master's degree in bioinformatics and the University of Exeter. And then she did both her PhD and postdoctoral work at the University of Cambridge. And Jenny has done extensive work on glycolipids in plants and plant cell wall and the role of the cell wall in selecting microbes. And I'm really excited to have her tell you a little bit about her project with the JGI. So with that, I'll stop sharing and just welcome you, Jenny, to giving this presentation. Thank you.
Oh, thank you very much, Trent. And now I am going to share my screen. And right. Hopefully you can all see my screen. Or are you still seeing presenter mode?
You're in presenter mode.
Awesome. All right, just get that sorted. Perfect. Great. So thank you very much for the opportunity to speak today. And I'm going to tell you about a project which we have been working on as part of the Joint Bioenergy Institute, which has really relied on this on a sort of integrated project with the JGI, so that we were awarded a couple of years ago. So we're going to talk about how we've been using integrating omics data to explore engineered phenotypes in the field. And this is a field of switchgrass, so switchgrass is what I'm going to be talking about today. But first, I'm going to introduce you to the Joint Bioenergy Institute, for those of you who are not familiar with it. It's a Department of Energy BER-funded bioenergy research center. It's one of 4 bioenergy research centers across the country. And our goal really is to develop biofuels and bioproducts derived from dedicated bioenergy crops. And to do this in an economic way, we use synthetic biology to engineer kind of every step of the process. So we go from plants, and we particularly work on sorghum, switchgrass, and poplar. We pull apart this biomass, which is essentially the plant cell wall, using methods such as ionic liquids to release simple sugars and also simple sort of other lignin monomers, which can be used by engineered microbes to make bioproducts and biofuels. And people have talked a lot about biofuels. I'm guessing most people are fairly familiar with it, but what do we really mean by lignocellulosic biomass and dedicated bioenergy crops? So today I'm going to talk to you about switchgrass. This is Panicum virgatum. This is a C4 perennial grass that's native to North America and has been discussed a lot as a potential dedicated bioenergy crop because of its ability to grow in fairly kind of challenging environments. And it's polyploid, which means it has a pretty large genome, 1.4 gigabases. There's a good draft genome sequence from the JGI. It's an obligate outcrosser. So this is a problem that I'll discuss later, which means that you can't self-pollinate. So this is hard for doing genetics in. And we're working with variety Alamo. This grows about 3 to 5 foot tall in the field, but it's transformable. So we can do genetic engineering in it. And it's high yielding. So even this variety here produces about 14 to 27 megagrams per hectare per year, depending on irrigation and nitrogen regimes. And because it's perennial, you can plant it in the field and then you can cut it back repeatedly, and across many, many years, you can continue to harvest it. And so for the plant cell wall, this is what we're really talking about. So all plant cells have a very thin primary cell wall surrounding them. But if you take a cross section through, for example, the stem, you see, so these are some water conducting cells. You see these thickenings around the outside, and that's the secondary cell wall. So not all cells have a secondary cell wall, but because they're so thick in comparison to the primary cell wall, the material you harvest is really dominated by this material. And the composition is determined both by the species and also the tissue type. But essentially, it looks like this. You have cellulose, which is a beta-1 4-link glucose, which forms these crystalline fibrils here. So decorating that, we have these hemicelluloses. These are a mixture of pentose and hexose sugars. And then in some cell types, we get lignin, this polyphenolic network that I'll talk about today. So what do I mean by lignin? Well, at the face of it, it's actually a relatively simple molecule. It's made from 3 major monomers. We have p-coumaryl alcohol, we have coniferyl alcohol, and synethyl alcohol. These are made within the cell and then exported to outside the cell, where they go through a process of sort of randomized cross-linking via reactive oxygen species, so driven by peroxidases and lactases. Now, because this is a randomized process, even though you have a relatively simple pool of starting material, you get this very heterogeneous polymer out. And so when these monomers are integrated, we get what we call H units, so p-hydroxyphenols, we get G units, these guaiacyl units, and syringol units. So people talk about lignin often in its ratio of GH to S, because that really controls the properties. Now, I will point out that other monomers can be used, and actually that is important, I'll come back to later in the talk, but these are the 3 major ones that we see. And one of our goals in JBEI was to make the processing of plant biomass into biofuels more economically viable. And lignin is a real hindrance to that process. So lignin kind of coats the outside, it makes access of enzymes to break down the sugars, makes it very difficult to get access to those sugars. And it's also a large amount of carbon in your biomass that it's very challenging to use. So can we make that lignin simpler to use, and perhaps can we decrease it? Now, this is not a new problem that's been asked. Reducing lignin is also important, for example, for animal forage, because it also hinders animal digestion. But through conventional breeding, if you decrease lignin too far, the plants simply just fall over and weaken. By using some engineering, we can actually do this in a cell-specific manner, which means we still get a strong stem. So, for example, our water-conducting cells are protected, but we can change this specifically in the fibers, which seems to allow the plants to grow normally. And the approach that is being used in this project was actually developed by my colleague Emmerich Udez and former colleague Dominique Locke, a paper that's published actually back in 2015. And it's a really beautiful strategy for trying to engineer lignin. So they introduced this QSUB gene, which is a bacterial gene, which converts 3-dehydroshikimate into protocatechuate. And this has 3 impacts on our engineering. So it produces, this protocatechuate is actually a really valuable precursor. So it can be used in making bioplastics. So by having a bioenergy crop that makes this precursor, you've already increased your downstream processing value. But shikimate is also part of this pathway in the plastid that makes phenylalanine. Phenylalanine is then moved out into the cytosol, where it's the entry point for the lignin biosynthesis pathway. So if you're reducing flux into phenylalanine, you're actually you can aim to reduce lignin biosynthesis. But the third thing this method does is by reducing shikimate. Shikimate is also a requirement for this enzyme here, HCT. So this is after the production of the H subunit, but it's needed for going further down the lignin pathway to make G and S. So this is what you can see here. By reducing shikimate, we expect to reduce flux through this specific part of the pathway. And indeed, that's what they reported. They found a decrease in lignin in the Arabidopsis. They found improved sugar release. They found increased protocatechuate. And in what lignin remained, they had an increased ratio of H to G and S. So far, so good. But no one's going to be growing Arabidopsis as a biofuel crop anytime soon. So one of our kind of task goals in JBEI is, can we transfer these technologies into dedicated bioenergy crops? So Emmerich has been working to transfer this to switchgrass, poplar, and sorghum. And today I'm going to tell you about our switchgrass data. So he ended up with 8 independent transformed lines containing this QSUB construct in the switchgrass. And in the greenhouse, these plants grew normally. They showed an increased amount of protocatechuate and a reduction in lignin. It wasn't as extreme as we saw in Arabidopsis, but it was certainly significant enough to be worth working with. And so our next steps were then, okay, now we've grown it in the greenhouse, we really need to demonstrate that this is functional in the field. I mentioned those concerns about loss of lignin weakening the wall. And we kind of said, well, we can do cell-specific engineering and that gets around those things. But there was a lot of concern, if you particularly talk to agronomists, that what you see in the greenhouse really doesn't reflect what you see in the field. So they need evidence that this holds up. And because these plants are obligate outcrossers, I also want to point out that to do a field trial, we have to actually split the plants through propagation. So we actually have to cut the plants in 2, and we have to move them into individual pots. So it actually took the full team to do that. And this is what you can see in our field trial. So this is our field up at UC Davis. It's so the folks at UC Davis are part of the Joint Bioenergy Institute. So this is them preparing the field. This is our plants planted out in 2017. They overwintered, and then in 2018, we could start doing our analysis. And we used a randomized block trial, which you can see here. And what that means is that for every block that you can see, we have 30 plants in 3 rows of 10. And we have 4 replicates of each and 3 independent lines of this QSUB. So we went from 8 to 3. Plus we have extensive control, these Alamo control as well. And this is a drone picture you can see of our field trial. And I'm hoping you can see that some of these blocks seem smaller and some of them seem bigger. And so when we actually did our wet chemistry analysis, we were kind of surprised and a bit disappointed to find that in these QSUB lines, the lignin content was actually unchanged compared to the wild type. So we weren't recapitulating our greenhouse phenotype. The protocatechuate, we saw a bit of an increase, but it was not as big as we would have liked or hoped for. But the QSUB lines were about 30% bigger. So we had this huge increase in yield. And yield is great because more biomass is great for the farmer. That's what they're looking for with the same inputs. And so our question was, okay, why did introduction of a single QSUB gene cause such a big phenotypic difference between greenhouse grown and field grown plants? And I should say that we have seen the same yield increase across now multiple years and in multiple field locations. So we're pretty confident in it. And so this was we had already started thinking about the idea of trying to understand what that transfer between a greenhouse grown plant and a field grown plant looked like. And so we approached the JGI and explained our experimental setup and submitted a BRC project to work with them, where we took this setup. So we've got these 3 QSUB lines. We've got these clonally identical plants. The plants in the field and the plants in the greenhouse were propagated by splitting. So they are clonally identical. And then we took samples from the 2nd and 3rd harvest of 2018 and actually of 2019 as well. And we developmentally matched them to some plants grown in the greenhouse. So we took the same tiller age of both. And then for the 2018 samples, we've done transcriptomics and metabolomics on 1 harvest and transcriptomics on a 2nd harvest as well. And we've also done wet chemistry, so lignin composition and monosaccharide composition as well. And I should also say that we were pretty new to doing this type of work. We hadn't done any type of metabolomics before, or particularly not untargeted metabolomics. So we worked with the JGI to do a pilot experiment because they hadn't done switchgrass before. So we could work out the number of replicates we needed for the metabolomics. And we settled on 8 as giving us a good data quality. And that's what we can see here in the overview. So first thing is, how do you deal with this much data? There's a huge number of features. Sharon Greenbaum has developed this way of us looking at the data as an overview. And so here you can see our transcriptomics and metabolomics. And one thing is that we actually found that the samples cluster differently in different omics datasets. And what I mean by that is in the transcriptomics, the separation first was by genotype. So we have all the QSUB samples over here and all our controls over here. And this was different in metabolomics, where it seems that location, so greenhouse, dominated over the field. And then in the transcriptomics, the second most important thing was the location, whereas in the metabolomics, it was the genotype that's next. So you can see this clear clustering of this is our control over here and our QSUB lines over here. And nicely for us, we only had 3 independent transformed lines of the QSUB in the field. And they had, in sort of large scale commercial field trials, you would have many, many more. But actually between our 3 lines, they didn't cluster out along those 3 lines. So we are pretty confident that these 3 independent transformations are equivalent. Now, there's lots of different ways of dealing with this data set. And one way is to start with just the transcriptomics. So we could look at, for example, what gene ontology terms are upregulated in the field versus the greenhouse. And so this is what you can see here. And we can start to pick, there's actually very few, these are the only significantly upregulated processes we saw in the field versus the greenhouse. And so, for example, we can see that in the field, we get an increase in all lines in the cellulose biosynthetic processes. So this is our control and our 3 QSUB lines. But we actually only see this category of plant cell wall organization and biogenesis here that you don't see in any of the QSUB lines. And the thing that really interests me is that the response to water and phosphate starvation is significantly upregulated only in the QSUB lines. And one way of thinking about the plants being able to grow bigger is that they might be more resilient to stress. They might be better able to deal with the stresses of the field versus the relatively comfortable life in the greenhouse. But I'm just going to switch over to metabolomics. Trent's actually done a really beautiful job of explaining what we're looking at here. So I'm going to skip over this and just point out that for those, so we got a huge number of metabolite features back. And for many of them, we could actually then use the online database to do spectral matches to actually confirm identity and be very confident in what we're looking at. So here you can see tyrosine, and you can see that our mass spectra correlates very well with that found in the database. So we can be very confident that we can assign these types of features to being, say, tyrosine. But as Trent mentioned, it's very, very complex. So where do you start looking at the data? One way is that you can take an almost kind of targeted ab initio approach, like start thinking about where are my lignin related compounds. So we did this to start with. We went through and sort of pulled out the lignin precursors. A lot of them standards exist, which makes it a lot easier. So this is p-coumaric acid, for example, and we get these types of box plots out. So you can say that in the field versus the greenhouse, we have an increase in this p-coumaric acid, but in all 3 QSB lines, this is enhanced. And since this is the H-lignin precursor, we might expect that because that's above the blockage where the lack of shikimate sits. But as Trent said, a lot of features don't have spectral matches. So how do you go beyond this? So one way is to make use of this GMPS network they mentioned, where we can take these metabolite features which have very similar structures. So we can kind of make some predictions that if metabolites have very similar structures, they're likely to be part of the same pathway. And then you can paint on top these correlations with gene expression. And this is here we pulled out all the switchgrass genes which are predicted to be involved with the lignin biosynthesis pathway and painted them on the top. And we see these features that correlate very closely. So does that really mean anything? Well, you can actually go in and check. So correlations do seem to support known relationships. And this is one example of actually a very strong negative correlation. It's at the strongest hit we had. And here we've got, this is a metabolite correlation with the gene score, the Z score of both. And here you can see it's shikimate O-hydroxycinnamyl transferase, which is this HCT gene, and shikimic acid. And it's got extremely strong negative correlation. And it seems this makes complete sense. So HCT uses the shikimic acid to make its this p-coumaryl shikimate. So anytime they're present, it's going to get converted. And we can do this for a number of different components of the lignin pathway. But how do you go beyond that to really thinking about this integrating these metabolomic and transcriptomic data sets? And Sharon Greenblum and Ben Bowen have developed these really beautiful systems for pulling out this data. And this is just one of this lignin-related subnetwork that I mentioned. And what we could see here was two clusters. We have this cluster which circles around this CYP73A, which is the C4H gene up here. And the circles are genes and the diamonds are metabolites. And this pinkish color means that they are overaccumulated and green means they're underaccumulated. And so around this CYP73C4H, we see a cluster of metabolites which are overaccumulated. And then around this HCT gene again, as well as the C3H down here, we can see this cluster of metabolites that are underaccumulated. And again, this makes sense. C4H is up here, so we are kind of above this blockage that we've introduced in the pathway, and HCT and C3H are below. And this was specifically greenhouse-grown plants versus the QSUB10. But when we look in the field, we actually, in our field data, so this is exactly the same plant, but in the field, we've got this, the field-grown control with the field-grown QSUB. Again, we see this top cluster with the C4H with these overaccumulated metabolites, but hopefully you can see that down here now with the HCT and C3H, these metabolites are now overaccumulated. So something strange is happening in the field where we seem to be kind of overcoming that blockage and getting this, getting the lignin still being synthesized and produced. And in particular, I was kind of curious to note that one of the metabolites that we see a big change in is L-tyrosine. The reason that's of interest is because in 2016, it was reported, so after we kind of started this project, that in grasses specifically, so not in Arabidopsis, there is a different route to producing lignin that doesn't go via phenylalanine. In fact, it goes via tyrosine. So there is a gene, a tau, which converts this tyrosine to p-coumarate, which then continues down the lignin pathway. And so what it seems to be that in the field, we may have a different emphasis on these different pathways. We're still not clear how it's dealing with the lack of shikimate, but it's something we want to continue to explore. So just to wrap up, we've got a lot of future work. This is a huge data set, and we honestly think we could spend years exploring different parts of it, and that's a pretty exciting thing to have access to. But in particular, we want to kind of focus on those clusters specific to that field versus greenhouse effect. So what's really driving this yield increase that we're seeing? Because the lignin work has been slightly sort of more straightforward to analyze because we had some sort of fundamental understanding of how the lignin pathway was synthesized. So hormones and transcription factors might be good clues to look for, as well as future additional engineering targets for a QSEB switchgrass version 2. Now, one of the dreams in synthetic biology is that you actually do what we call this design, build, test, learn cycle. So you do your engineering, you take your plant, you learn from it, and then you go again. Can you improve on what you've already made? Now, in microbiology, that can be a very quick turnaround, those design, build, test, learn cycles. Plants, it's much, much, much slower. But by having this much data on something like switchgrass, we can actually make some predictions about where we can engineer and then the sorts of conditions we should grow the plants in to see sort of field-like phenotypes. So for example, should we be targeting the tau enzyme, the tyrosine part of the lignin biosynthesis pathway as well? We also plan to do some targeted metabolomics to confirm identity. And we would like to explore some other of the phenylalanine dependent pathways like tannins and anthocyanins. Is one of the reasons for the change in the lignin emphasis on phenylalanine versus tannin, something to do than tyrosine, because of the requirements of for phenylalanine and other pathways when it's grown out in the field. And finally, I'm really a biochemist at heart, and I'd like to do some feeding experiments to test 13C tyrosine versus 13C phenylalanine in QSEB switchgrass. And that's really challenging to do in the field because we'd like to see that difference between a greenhouse type field-like conditions. But this is something that I think might be possible at LBNL in the future. We've just taken delivery of the first ecopod, which is a fabricated mesocosm, where we can, under very controlled conditions, grow plants like over a full life cycle and sort of apply field-like stresses. So finally, I just want to thank the team who's worked. There's been a lot of people, as you can guess, but particularly in my team, Kavitha, who's kind of led the field work, Xiaozhan, who's doing a lot of the transcriptomic analysis, and John Diab, who has taken on, especially during shelter in place, a lot of the metabolomics analysis. And Emmerich, who really has driven this QSEB work, and the whole feedstocks team. But I would also particularly like to thank Ben, Sharon, and Catherine, because they've really helped us at every stage of the project, from doing that initial pilot analysis to work out how many replicates we needed to do the switchgrass analysis and metabolomics, but also to doing all this data analysis, and particularly these integrated omics tools. And with that, I'll stop sharing and thank you for listening.
Thank you, Jenny. That was a great presentation. We have some questions that have already started coming in about your presentation and just in general. So if I could get Trent to sign on here too, I'd like to start asking some of these questions. Okay, well, so the first question I think might be for you, Jenny. If you could talk just a little bit more about what tools you were using to integrate the metabolomics and transcriptomics.
Yeah, sure. And maybe Ben can chip in as well. So this was something that's completely new to us. So Sharon and Ben Bowens were able to put together actually this beautiful Jupyter notebook, which can take our samples and has lots of different tools integrated in to produce these outputs that let us explore the data. Ben maybe can speak to the details a bit more because that's really not our expertise.
Sure. The codes that we're using right now are mostly Python and R scripts that are part of these Jupyter notebooks, as Jenny mentioned. But because it's not that uncommon to have paired data sets with transcriptomics and metabolomics, we're aiming to make them a core capability for the platform. But we also use a software package called Magi that Trent mentioned. And Magi is more of a mechanistic linkage to try to find a rationale for linking the metabolites and genes in addition to just correlations. And yeah, so a variety of tools that we use, from custom scripts to packages for gene ontology and other set enrichments to Magi.
Great, thanks Jenny, and thanks Ben. I have another question here, which was about just asking to talk a little bit more about the technical limits of applying an untargeted approach to the rhizosphere, just in terms of limits of identifying the molecules that are exchanged.
Limits in the number of samples, or the limits in the number of metabolites we can identify?
I think the question was about the number of metabolites you could identify.
Yeah, I mean, that really depends on the particular system. As we've described, we have 4,000 standards, over 4,000 standards that we use to annotate your samples. Typically those, by using that library, we can identify, several hundred metabolites. And then all the other molecules in your sample would have, less confident identifications. We call them sort of level 3 type identifications, just based on MS-MS, or in some cases, we might only be able to take a guess at what family of molecule they might be in. And occasionally we also find completely novel molecules that we can't annotate at all. So, you would get a table or two tables. One table would have all of the metabolites that we could confidently assign using standards. And then you'd also get untargeted output, which would be our hypotheses as to identifications of metabolites. And those are the ones where, if you really wanted to focus on a particular metabolite, it would be ideal if we could do some follow-on analyses with standards, et cetera.
Okay, thank you, Trent. We've had a number of different questions about the input material. So for folks who do sequencing, they, normally they would understand they need to extract their nucleic acids for sequencing. We've had a question about what is delivered. Does it have to be extracted? Can you use cell media, lake water, even raw rumen samples? What can they send?
Well, so we one thing about metabolomics is you can use it to analyze just about anything, but oftentimes there are interferences in the samples. And so then we have to come up with a protocol to separate the metabolites that you're interested in from the interferences. So just to give you an example, analyzing metabolites from a very salty sample, is particularly polar metabolites, is very challenging. And so sometimes we have to use extraction and solid phase extraction to be able to separate those metabolites. And frankly, sometimes it's just not possible. Like if you had a hypersaline sample and you wanted to detect some very particularly polar metabolites, that would be extremely difficult. But in general, we use liquid extraction with an organic solvent. And in general, and I saw there was a question about, is there like a type of liquid extraction that is best for sort of overall? And we found that methanol works very well. But one issue with methanol is that that salts can be quite soluble in methanol. And so sometimes we have to use a slightly different approach for particularly salty samples.
Yeah, I can just to add that for our experience from switchgrass, so we worked with Catherine Louis in Trent's group to work out how many samples we needed for these reproducibilities. So we sent them samples, and all we did is we sent them frozen ground leaves that came straight from the field. And that's why we settled on doing eight technical replicates. That gave us sort of very nice data, but that allowed us, but yeah, it was just frozen ground tissue straight from the field, which is pretty easy for us to prepare. About 30 milligrams, I think, or something like that.
this is why we perform these pilot studies, is because we don't want you to go through some huge experiments and then send us the samples, and then we find out that there's some issue with salt or surfactants, or something, you don't have enough sample to really detect the metabolites that you're after. One thing I just want to mention to everybody is that it is incredibly useful if you do have at least one hypothesis about a metabolite or a group of metabolites. Like Jenny here, she was interested in lignin biosynthesis. So that gives, lets us know, hey, okay, aromatic compounds are important, so that when we perform a pilot experiment, we can make sure that we're seeing those classes of metabolites. So the more, oftentimes people use metabolomics for hypothesis generation, and that's totally fine, the untargeted approach, hypothesis generation. But if you do have a hypothesis, that allows us to really de-risk your study.
Thanks, Trent. Yeah, it definitely doesn't sound like a one size fits all, and there's some degree of customization and on each project. I think more so than in a lot of our sequencing projects. I've had some questions here about a little bit more about there's this kind of a series of questions about the how results are provided to users, what type of support is provided, and are there any sort of training opportunities?
Yeah, I'm going to let Ben take that question in terms of just sort of our routine output, and then also opportunities to engage with him on helping with the analysis and learning how to use some of these tools.
Sure. So it's not really the one size fits all answer. Again, like Rick said before, the analogy for extractions, the same with analysis. Some of the users have request analyses that are literally looking for a single compound, a product from DNA synthesis reactions. Other users are more in the hypothesis generation realm, where we're doing a deep exploration of thousands of features from an untargeted experiment. And then most users really tend to gravitate to our targeted pipelines, the targeted polar metabolomics pipeline, where users will typically get back a table with a few hundred identified features, anywhere between 50 and 300 would be the range. And those identifications are ranging from extremely high confidence, where you have a very nice clean signal with a retention time match, an M/Z match, and a fragmentation match, and no confounding signals at all, down to lower quality identifications where it's much less uncertain, and all we can say is it's a hexose or it's a sugar. Those tables are provided to users where they get intensity data, figures that they can provide as supplementary material. And we try to conform to these metabolomic standards requirements, where we provide tables with these evidence tabulated in a way that they can easily publish that data. The untargeted pipeline is completely different. They will get back a table of features, which is just a density of signal at an M/Z and retention time. And for the majority of features, they'll get back a fragmentation spectrum for that feature. And then their data will be run through the GNPS pipeline. So people mentioned training opportunities. There were many questions about training opportunities. The GNPS pipeline has numerous YouTube videos and web-based documentations and users support. So it's a great place to start if you're getting into metabolomics analysis, to just begin watching some of these YouTube videos and trying it out with some data. They have a lot of public data that you can explore. And yeah, hopefully our users come away from a metabolomics project with a deeper understanding.
Yeah, one point I want to make that is we're very proud of for the metabolomics, the targeted metabolite analysis, is that we actually go to the extra effort of manually inspecting all of your data for these most confident identifications. One of the challenges with metabolomics is that it's pretty easy to confuse a fragment of a particular metabolite for another metabolite, adenosine and adenine, for example. And we would hate for you to go write a paper because you have some interesting finding about what turns out to be the fragment of something else. And so we do go through and spend a lot of time curating your data before we send you the targeted analyses. But that takes a lot of effort and time. But we have not been able to find a way to eliminate that step. We've been able to speed it up and make it easier for an expert to go through and annotate your data. But really, it takes that deep expertise in all of the different types of experimental errors that can happen to make that curated table.
Thanks, guys. I have another question here for you. So there's obviously an important connection between enzymes and metabolites. Are there any plans to connect proteomic data with metabolomic data?
I'll just quickly say a few words and then I'll let Ben maybe expand upon it. So his Magi software works with any type of FASTA file. So basically you upload two different tables. One is a table of annotations for FASTA file, and then the other is annotations for metabolites. And then the Magi will determine the consensus between the two using this chemical reaction network. So it works for metagenomes, transcriptomes, genomes, proteomes, etc. Ben, do you want to expand on that, or did I do an okay job?
Yeah, sure. And we can also mention that there are other DOE users facilities like EMSL that have a long history of doing proteomics.
Yeah, that's a great point.
So I mean, you have a combination of ways to think about enzymes and proteins. Magi will try to use chemical reactions and enzyme promiscuity and homology to reference databases to make those links to transcriptomics data, metagenomics data. Many of the data sets we get are rather large experiments, or like medium to large experiments where there's dozens to low hundreds of conditions. So the comparative analysis can try to link speculated protein sequences with metabolites through these informatic means. But yeah, I think doing proteomics in addition to transcriptomics and sequencing would be just a great idea. I used to do proteomics a long time ago. It's a terrific technique. It's often correlated with transcriptomics, but not always.
Yeah, and I should mention that one of the proposal types that you can apply through is this FICUS program, where you can ask for proteomics at EMSL and then metabolomics through JGI and transcriptomics, et cetera. So there really are a lot of capabilities that you can assemble in terms of systems biology through the two different user facilities.
Yeah, and that FICUS proposal, we don't have the deadline set. I'll have a slide up on it again, but it'll be in the spring of 2021 for when you can apply to this joint EMSL JGI proposal. Okay, we're getting a lot of questions. I am looking at the clock. I can tell that we're not going to be able to answer them all, but I'm just have one last question for you, Trent, then before we close down. And I want to let folks know that we're collecting these questions, and when we archive this, we're going to try and get back to you and answer these questions later if we couldn't do it now. Okay, so quick question about about the proposals. Are there caps on the numbers, or what are the caps on the numbers of samples that can be submitted for screening?
Oh, great question, and I'm glad that we got to that one. So for the functional genomics and new investigator proposals, those are more limited. So currently those are limited to 50 polar metabolite analyses, so 50 samples, and 150 non-polar metabolomics analyses. For all the other proposals, it's much larger. So it's 200 polar samples and 500 non-polar samples. And those exact limits, are something that can be discussed. If there was a really strong rationale to go a little bit above that, then that's something that could be considered. But those are generally the limits.
Okay, thanks, Trent. So I think that's going to do it for the questions. If you had any last thing you wanted to say, Trent, any last point to reiterate, go for it. Otherwise, we'll close it up.
Sure. Well, I mean, I just want to thank everybody again for attending this webinar. And then I also want to thank Rex for hosting it, and Jenny for an excellent talk. I'm always accessible, so you can email me if you have any questions about the program or applications. And I know that there were several questions about, can you analyze this or that? Those are great questions to ask. If you're thinking about a proposal, you can reach out to me and I can help you sort of think through whether this is a good, the JGI Metabolomics program is a good fit for your needs. I just mentioned I'm a really active scientist and I'm really passionate about this. And so one of the reasons why I'm so happy in this role is that it gives me a chance to explore new applications. So I'd love to hear about your interests and the kind of questions you're trying to ask. So with that, I just thank everybody again for attending.
All right, thanks a lot, Trent. I appreciate that. So I just wanted to take a last second to put this slide up again. This pathway up at the top, this is where you can go to find more about the user programs that we offer. Again, you can request metabolomics through these different calls. The next one is the functional genomics at the end of January, and then the new investigator call. The difference here is with the new investigator is this is for folks who have not been a lead PI with JGI before. It's restricted to just them. So that can be an opportunity if you haven't worked with us before to start that, to get your foot in the door. And then after that, we don't have the date set, but in spring 2021, we'll have the call for the FICUS proposal and then our annual large-scale CSP. Okay, well, thank you very much, Jenny and Trent and Ben for answering questions and the nice presentations. I'm sorry again to those folks whose questions we didn't get to. We're gonna, like I said, we're gonna try when we archive this, we're gonna make the video available and also see if we can answer those questions online after the fact. A quick note, we're gonna send out a survey momentarily, and we would really love to hear back from you and get your feedback on how we can do this better and provide more information. You can also stay in touch with JGI in general through sort of the normal social media outlets here. And you can also hear about some interesting stuff we're doing on these podcasts. Okay, well, that's it for today. Thank you very much for attending. Please reach out to us if you have any questions, and have a great day. Thank you.